First, I just wanted to thank the organizers for getting us all together in the room. It's really very important for researchers to come together with clinicians and families to really guide our research and make sure that we're asking the right questions and focusing our efforts on what's important. I don't have any disclosures for this presentation. I'm going to skip the objectives because you have those. Hop right to it. So really the dream is that you would get your SCN2A variant and we would be able to tell you, here's the drug that's going to make everything better. And that's what precision medicine promises. It's not exactly where we are. But there are ways to research to try to get there. And some of the tools that we are using include things that you've heard about during this meeting. We can look at several different levels of analysis, including heterologous cell models, where you introduce the channel with a variant into a cell that normally doesn't express the channel, and you can see how it's broken. You can look at iPSC models that we just heard about, and you can differentiate those iPSCs into neurons in a dish and look at how the mutation affects neuron function, or you can make an organoid and look at how the neurons talk to each other. Or you can do kind of what I think of as the more simple version, where you let an animal make a brain for you, and then you can study both the whole animal, or you can take neurons out and study those and look at how they affect neurons, or you can look at a slice, which is sort of akin to an organoid, and look at how the neurons talk to each other. All of these are very valid approaches, and they answer different questions. So it really depends on what your question is, which model is the best system. And one of the big questions is how do you integrate across those different systems? And that's been a big goal for the U54 Center that you heard Dr. George talk about yesterday, is we want to look and see what are the salient pieces of information from each of these systems that can get us to better treatments. And so we have 3 projects. One is looking at the high throughput evaluation in heterologous systems. Another is looking at iPSC-derived neuron models. And a third is looking at mouse models of variants. And what we want to do is look at some of the same variants across all these models and try to put it all together and synthesize what the useful pieces of information are. So you heard about this automated system yesterday from Dr. George. It lets us look at a lot of variants under the same exact conditions, which is actually really important because electrophysiology, if you vary the conditions just a little bit, one investigator might see one result, another investigator sees something slightly different. So if you can compare a whole slew of mutations under the exact same conditions, you can make comparisons relative to each other. And you can do this in a high throughput way. And that really helps us focus on what are the different classes of mutations. And we can try to put them into buckets, the gain of function and loss of function that you've heard a lot about. And there are different criteria that make them fit into these 2 categories. There maybe are limitations in trying to put them only into these 2 buckets, but it's a good place to start. The other thing that we can learn is what are the representative mutations that we can take forward to these more complicated models. So this is just one example from the high throughput automated recording. This is kind of how I picture it. So you can see a couple of different patterns. Your representative traces from wild type, and there are variants that look very similar to wild type like this one. There are some that Dr. George presented yesterday that the morphology looks different, and these turn out to be gain of function mutations. And then there are others where you have less current, and those would be classified probably as loss of function. But we shouldn't forget all that we've learned over the last many decades of ion channel biology research. Dr. Catterall gave us a nice introduction to that. And these channels have been studied for a long time, and we know an awful lot about them. And K1422E is one of those variants where we know a lot about it. So this probably isn't the total answer for that variant. So in the structure of the channel, we know that there is the voltage sensing domain, and there's the pore forming domain. And if you turn that on its head and look down the center of the pore from the outside of the channel, there are those 4 selectivity residues that line the channel pore and determine selectivity for sodium and keep out other ions. And K1422 is one of those critical residues. And one of the ways we know that, oops, it's a little fussy, is by doing studies where you do site-directed mutagenesis and you look at the effect on channel function. So for K1422, in all sodium channels, this is a K, but in calcium channels, it's an E. So back in the early 90s, they mutated this K in the brain channel, and actually this was in brain 2, which is SCN2A. This happens to be a rat brain channel, but it's very similar to the human channel. And what they found is you make it into a calcium permeable channel. So you basically convert a sodium channel into a sodium channel into a calcium channel by doing this change. So Chris Thompson, who's a scientist in the George lab, recapitulated this with the human SCN2A channel. So if he records under standard conditions, you don't see very much sodium current. But then if you look under sodium-free conditions and allow calcium to move, you can see that there is a calcium current. And so this is actually a change of function. It's not really a loss or strictly loss or gain. It's really a change, and it has a new function. And so this is really hard to predict what the effect would be in the nervous system. So we thought this would be a great opportunity to make a mouse model. So we use CRISPR-Cas9, which really makes it a lot easier to make knock-in mutations. So we just changed a single nucleotide in the mouse SCN2A gene, which is 98% identical to the human gene. And we got viable and fertile heterozygous animals. That's important because then that means we can breed them and propagate them and study them. The homozygotes die within a few hours of birth. That's exactly what you see with an SCN2A knockout mouse. And it looks about the same. So there are some features that might overlap with the knockout. We do see a normal level of SCN2A mRNA as well as protein. So that tells us that the DNA is converted into RNA, the RNA is converted into protein. The other really helpful thing about this mutation is it also renders these channels resistant to TTX. So you get this huge shift in the IC50, and that means you can isolate the mutant channels in neurons because all wild-type neuron channels are sensitive to TTX. So these are just representative traces. On top, we have wild-type. There's TTX-resistant current is very little. That's really chloride conductance. And the TTX sensitive is all the brain sodium channels. But with the K1422E, you have some TTX resistant current that you can see, and then you can isolate that and look at it. There we go. So just if you focus your attention over here on the right, Chris recorded showing the TTX resistant current blown up here, and you can see wild type has very little current, but with the K1422E mutation, you can see that there is some TTX resistant current. Oops. So what does that do in the brain? We do things that are similar to what you do in the clinic. So one of the things we do is video EEG monitoring. And this is done by Sunita Misra with some help from Nicole Hawkins. And it's really in the preliminary stages, but there are definitely some interesting patterns emerging. She identified some epileptiform discharges, which are isolated spike and slow wave discharges. And these are just representative of 3 different mice, so it seems to be a reproducible phenotype. Another thing that we can do that's a little bit faster is we can look at seizure susceptibility. And for this, we use fluorothil, which is a GABA inhibitor. And we look at how long does it take to go from the first seizure sign, which is a myoclonic seizure, and also look for the generalized tonic-clonic seizure, which is the final event in this sequence. So, and this is mislabeled, this is latency to the myoclonic jerk. It's no different between the mutant and the wild type, but there's actually a delay in the threshold for the generalized tonic-clonic seizure. And what this tells us is that the animals are actually a little bit resistant to seizure spread, which might suggest that you would see focal seizure, or you would expect focal seizures over generalized seizures, at least in the mouse model. And another thing that we've been doing is a lot of neurobehavioral phenotyping. And this is all being managed by Dennis, who's also in the audience, with some help from a couple of technicians in the lab. And we've done a bunch of different tests, but I'm just going to show you a few where we see some interesting outcomes. So the first is an open field test. And this is really a test for general motor function and activity level, as well as looking at anxiety-like behavior. So really, we just put the mouse in a box in a brightly lit room and let them explore. And their natural tendency is to stay towards the walls. And this is just a trace of all the movement over the course of 30 minutes. The wild type mice will stay pretty close along the walls. And what we found, in fact, was that the KE mice tended to spend a little more time in the center, even though the distance that they travel is about the same. So that suggests that they have either less anxiety-like behavior, or at least their behavior isn't inhibited by this normally aversive condition. So with behavior in mice, you never trust one test, you always do multiple tests. So I have maybe another one. Oops. Okay. So zero maze is another test that also looks at anxiety-like behavior. And in this case, the mice are on a platform that's a zero, and it's up pretty high off the ground. And again, it's in a brightly lit room. These open arms are pretty aversive, and these closed arms are a safe area, and the mice will tend to stay in that safe area. So you're tracing from a wild type mouse. You can see he pokes his head out here a little bit, makes one track across the other open arm, but basically stays in the closed arms most of the time. So again, we see that there's a difference between the wild type and the mutant mice, with the mutant spending more time in the open arm. And again, they travel the same distance. And then a third test of this anxiety-like behavior is to look at, again, this light-dark difference. And in this case, there's an arena that has a brightly lit area, and then there is an enclosed area that's completely dark. And you can look at, again, total distance traveled. We don't see any differences. You can look at the number of times they pass through this little door between the two arenas. We don't see any difference in that, but we see that the KE mice spend more time in that open space. So again, their behavior is a little less inhibited. So then switching to a different task, this is the beginning of looking at social behavior. One of the important things in mice is that a lot of their social behavior is driven by olfaction, so smell. And so we need to test their olfactory ability. Can they smell things first off? Because if they can't smell, they can't do the tests. And this is a little bit of a hybrid, this olfactory habituation, dishabituation test, because it tests ability, but it also can give you the beginnings of looking at social deficits. So what you do is there's a cotton swab that has a smell placed on it, and it's introduced once. That should be really interesting if it's a new smell. It's introduced a second time, it should be a little less interesting. And a third time, it should be boring. They shouldn't pay much attention to it. And so this is done first with non-social smell. So we start with water, which is just a neutral. And you see that you get a little bit of interest because that's a new object, and then they habituate pretty quickly. And then it goes in a sequence of almond extract, banana extract. And what you can see is that the wild type mice, you get this large increase. So this is the dishabituation, and then you get habituation over the next two introductions. In the KE mice, they have a response, it's blunted. And then when you get into social odors, it's broken down by same sex and opposite sex. Their response to the same sex is actually fairly substantially blunted, but opposite sex seems to still be pretty interesting, which is probably why we don't have a lot of problems with breeding of these mice. But it's a little bit of a conundrum, because in some ways we're getting a signal that there is potentially a social deficit, but we're also getting a little bit of a hint that there may be an issue with olfaction in general. The fact that they can do it a little bit suggests that maybe they can collect the information, they just don't process it quite the same way. So what that means is Dennis has more work to do. So there are a whole bunch of social interaction tests. One of the classics is a 3-chamber test, and you may hear some of that maybe in Kevin's talk. But it's a very standard test that looks at sociability and social preference for new friends over either boring objects or friends that you've known for a long time. There's also more pure tests for olfactory ability, where you make them a little bit hungry, and then you actually bury a little cookie in their bedding, and if they can smell really well, they should find it pretty quickly. So that's something that we need to do. In addition, we've been doing a lot of recording of the animals just in their home cage and social groups, and that can be used to look at sociability just in their natural environment, as well as looking at things like repetitive behaviors, and what I think of as the mouse version of activities of daily living. There are things that mice do in their normal life, and I think we could really harness some of that data to try to see what activities need more follow-up testing, and what seems to be the most salient things for us to look at. So just to wrap it up, I hope I've convinced you that looking across these multiple different levels of analysis is useful. We can see from heterologous systems that there's a change of function with this mutation. And when we look at ex vivo neurons that are taken out of the mouse model, we do see that same change of function. So that seems to track very nicely between heterologous and neurons. And in the mouse model, we do see epileptiform discharges. We do see this kind of interesting and maybe counterintuitive resistance to seizure spread. And we also see neurobehavioral differences, some of which overlap with the SCN2A knockout mouse. So it's a little bit of a hybrid phenotype between the more gain of function epilepsy and loss of function autism. So I think making models, particularly for these mutations where it's difficult to understand what the effects in the nervous system might be, would be really valuable. And then they can also serve as platforms for testing treatments. Oops, just want to make sure. I think I tried to cover people on the way. But these are all the people who contributed to the work. In addition, we're being advised by Jill Silverman at UC Davis on some of the neurobehavioral testing. She has a lot of expertise in autism phenotyping in rodents and our funding sources. And now Sunita is funded by Families SCN2A. So that's very exciting. And we're hoping to continue and learn a lot more and move a lot faster. Thank you.